We'll start with today's webinar. Haystack Analytics welcomes you all for today's webinar. With the new series, we are starting a webinar with Fighting Drug-Resistant TB with Next Generation Sequencing, a Paradigm Shift, which will be presented by Dr. Anirvan Chatterjee and moderator here, Dr. Chaitali Nikam from Haystack Analytics. Let's begin with about the organization and about today's speaker. Haystack Analytics, a health tech company based at IIT Bombay, Mumbai, on a mission to bring genomics to the forefront of healthcare. Our tuberculosis genomic diagnostic solution, recognized as a top innovation by BIRAC and launched by the Prime Minister of India, showcase our commitment to healthcare innovation. Our flagship product, Infection, aligns with WHO's Sustainable Development Goals, driving us to make personalized therapy accessible to all. Our vision is to transform healthcare through genomics. Our focus is to fill the gaps in healthcare by creating scalable solutions to benefit patients and the healthcare ecosystem. Our tech partners include Intel and GE. We are supported by NTEP, BIRAC, CAHO, Nidhi Prayas, Sign, and many more. Our partners includes lots of majority tertiary care centers and the hospitals and renowned laboratories. Few of them are listed here. About Anirvan. Dr. Anirvan Chatterjee is CEO and co-founder at Haystack Analytics. He has led his organization to achieve remarkable success in genomics, intellectual property, and healthcare, and in under 4 years, the company has become a major player in genomic solution. Despite challenges in deep tech, Dr. Chatterjee created an organization which can not only create significant IP in genomics, but also brings value to the healthcare ecosystem. It is a momentum undertaking, taking deep understanding of the science, technology, and the team. Few of the awards and allocates are listed here. Dr. Anirvan is a Gates Foundation Fellow at Keystone Symposia in Cape Town, Travel Fellowship Grant which has been given to him for 44th Union World Conference to be attended at Paris. He is a Fellow for J.N. Tata Endowment, Tata Independent Grant for Education and Research. He is Novozymes Henning Hock Larsen Foundation Exchange Fellow and has completed his postdoctoral fellowship at Oxford University. I request Dr. Anirvan to take over and start with the presentation. Over to you, Dr. Anirvan.
Thank you, Dr. Chaitali, and welcome everyone to this webinar. As told by Dr. Chaitali, we are restarting this webinar series of TB whole genome sequencing, and we wish to connect with all leading pulmonologists and clinicians who are dealing with tuberculosis patients in India. For the uninitiated and also for just setting the context of the webinar, tuberculosis is a disease which is caused by Mycobacterium tuberculosis. While there are several kinds of tuberculosis and its etiology, primarily we talk about pulmonary tuberculosis and extrapulmonary tuberculosis. Both of them have their own challenges. While pulmonary tuberculosis presents the biggest challenge in terms of transmission and the spread of the disease, extrapulmonary tuberculosis in places like India and other places in the world are probably considered as one of the most important agents in PUOs. These statistics are not new to many of us, but it is only useful to remind us from time to time again what are we up against. On very conservative scale, in India alone, there are 2.64 million people infected with tuberculosis. Almost 4.5 lakh people die of tuberculosis in India annually. There are a large number of multi-drug resistant cases. There are actually cases of pre-XDR and XDR. While we do share a large burden of tuberculosis, what is more alarming for India as a country and the rest of the world is the increase in the drug resistance of tuberculosis, which can derail any of the current efforts made by the world's scientific and medical community to eliminate tuberculosis. And these are some of the things that I'm going to talk about today, and how can we do our bit in the fight against tuberculosis, in fact, in the fight towards elimination of tuberculosis. While it is very clear that TB itself is a disease which is extremely complicated to deal with, not only for the clinician, but also for the patients. I think the focus of the entire efforts of elimination of TB is largely in two parts. One is that of latent tuberculosis, and the second is that of drug-resistant tuberculosis. And we are focusing exclusively on our ability to actually handle the problem of drug-resistant tuberculosis. So as I start with that, it is only fair that we make it very clear what are the different kinds of drug resistance in tuberculosis. We know of MDR, which is resistance to rifampicin and isoniazid, pre-XDR, which is MDR plus fluoroquinolone resistant, and XDR, which is pre-XDR plus bedaquiline or linezolid resistance. These are the primary definitions of tuberculosis, which also change as the epidemiology of the disease changes. But one of the things that many clinicians deal with in India is, of course, mono and poly drug-resistant tuberculosis, because there are set guidelines for MDR, pre-XDR, and XDR. But when we deal with patients who are neither of them, but there are key drugs which they are resistant to, it becomes increasingly challenging for clinicians to deal with. And these are some of the things that I'm really, we are really excited that these are some of the things that I think the world has the ability to work with, and the clinicians have the ability and the options to deal with. In terms of drug resistance in tuberculosis, when we talk about the current challenges, and as I was just saying, in pulmonary TB and extrapulmonary TB, once we understand that there is drug resistance in tuberculosis, the big question, and this is where there is a change of hands between biology and maths, because when we talk of primary drug-resistant tuberculosis, we're talking about a person getting infected with a drug-resistant form of tuberculosis, whether MDR, PXDR, XDR, poly, or mono. But the initial infection is from a drug-resistant form of tuberculosis. And the problem with the major problem with this form of tuberculosis is that the world has the ability to actually treat drug-susceptible forms of tuberculosis very, very well with the drugs which are available through the DOTS program, through the several national programs of tuberculosis around the world. Now, the problem is that if you are able to treat the drug-susceptible form of tuberculosis, those patients are not going to transmit the disease. And we are left with the drug-resistant forms of tuberculosis. If they are not treated well, they are the people who are going to, that is the form of tuberculosis that is going to get transmitted. And that is the big problem of tuberculosis, because we actually have a chance of eliminating tuberculosis. But what we don't want is we don't want to replace tuberculosis with difficult to treat forms of tuberculosis. Acquired drug resistance on the other side is very interesting, and this involves the part of TB which concerns drug compliance, treatment compliance, and that is where, if there is a suboptimal treatment for TB, there is a chance that drug resistance might evolve in the patient themselves. And that is where a large part of the work around the world goes around the DOTS, the VDOTS, and the compliance part, where we can imagine that given the high proportion of TB in groups of people for whom it becomes very difficult to have a very long compliant treatment, there's a lot of support that goes on here. Now, this data that is given here, where the NTEP in India misses 10% of drug-susceptible TB and 50% of drug-resistant TB. It is very interesting that it is actually better than the rest of the world, because the Harvard study once showed that almost 75% of drug-resistant TB patients in the world remain untreated in the population. And that is where primary drug-resistant tuberculosis is very scary, because it can transmit, and transmission of drug-resistant TB is one of the biggest challenges that there is, which is shown in the graphic to your right. Now, when we talk about our ability to handle drug-susceptible and drug-resistant form of tuberculosis, it becomes very clear that in this particular case, and probably in all cases in healthcare, but nowhere more pertinent than in TB, the ability to test and treat gives us unprecedented advantage in dealing with the disease at a patient level, at a community level, and at a world prevalence level. If we are able to test correctly and treat correctly, TB is treatable. In fact, with the right combination of drugs to a TB patient, the transmissibility of the disease reduces by more than 90% in just the first 5 doses. That is how important diagnostics play in the role of treating tuberculosis and preventing the transmission of tuberculosis. In this way, in this scheme, what happens is when we are looking at diagnostics, there are few diagnostics which are subject to the experience of the interpreter of the test, like, for example, clinical diagnostics and radiological diagnostics. And then there are very 0 and 1 states of definitive diagnostics, which are pathological and microbiological in nature. And all of us are aware of the microscopy, the solid, liquid culture, solid culture, and now I think molecular. And I want to stress on this a bit. While molecular methods in TB have revolutionized our ability to very rapidly detect the right form of TB and treat with the right medicine, I want to emphasize this. Tuberculosis is probably one of the earliest diseases for which traditional standard of care has been superseded by molecular diagnostics, whether it is the CB-NAT. It is worth mentioning that the way the world came into terms with COVID actually comes from the world's work in tuberculosis in terms of CB-NAT, in terms of LPA, and so on and so forth. And the obvious jump from molecular diagnostics becomes genome sequencing, because the basic science is the same. In molecular diagnostics, we are looking for mutations. In whole genome sequencing, we are able to find mutations. The difference is, in molecular diagnostics, we have to painstakingly create kits, standardize kits for each and every mutation. In genome sequencing, none of that needs to be done. We can actually sequence the entire genome of Mycobacterium tuberculosis from a patient and identify any known mutation without the need for specialized kits. And that's what we are going to talk about today. And the reason we have the ability to talk about it today, and we at Haystack Analytics are restarting this entire webinar series, is to bring to all of you the extraordinary work of scientists around the world. What has happened today, the WHO on a regular basis actually provides guidance towards how to use genome sequencing for diagnosing and treating TB. I'm not getting into each of these manuals, and I would encourage many of you to read them. But there are sections in here where it is explicitly stated that there are several antibiotics, anti-TB drugs, for which molecular diagnostics is considered as the gold standard. There are cases of drugs in which the drug susceptibility using liquid and culture methods are actually not considered to be the gold standard. That is how far the world has come in the ability to use whole genome sequencing. So what is whole genome sequencing? How does this process work? This is a busy slide. Stay with me here. We see here that The process is almost the same between a molecular method and whole genome sequencing. There is a patient, the sample is collected, the DNA is extracted, and from there on, there is a small change in the process. And that small change is, instead of putting it on a machine which looks into maybe 4 nucleotides and maybe 100 mutations, such as an RT-PCR or a CBNAT, we take that DNA and put it into what is called as a DNA sequencer. Now, DNA sequencing is the method by which the world over people identify novel mutations. Once those mutations are identified, they are put into RT-PCR kits. Once they are put into RT-PCR kits, they are being used for diagnostics. What people realized in the field of tuberculosis is that instead of going through this year-long, or actually 3 to 4 year-long process of doing whole genome sequencing of a large number of samples, identifying the common mutations, then designing the kits, then testing the kits, and then validating the kits, instead of spending 3 years for every new mutation to be found and then put into global circulation for detection, why don't we put whole genome sequencing as the method for detection in every patient? And with that approach, where we have come to today is that whole genome sequencing actually can turn out to be more affordable for diagnostics than molecular diagnostics. And this is how we were able to reach there. If you see, a CBNAT today is a fantastic replacement for microscopy, but then a CBNAT is a poor replacement for culture DST, because a culture DST can give a readout of 13 antibiotics. A CBNAT plus an LPA can give a readout of 7 to 9 antibiotics, and that too not completely. And that is why, while CBNAT is a fantastic method for detection, confirmation of tuberculosis, it is not the ideal method for diagnosing TB, especially if we are suspecting drug resistance in a patient. Just to walk you through this, this is the algorithm, and I will come back to this in a bit. But this is the algorithm that the world is following today, where we take a TB patient, we do a CBNAT if rifampicin sensitive, then first line LPA, if rifampicin resistant, then second line LPA. And after all of that, the patient still needs to undergo culture DST because of the universal DST norms that the WHO has set out. And this entire process of discovering the 13 antibiotics, from which we can choose the right concoction, from which our clinicians can choose the right concoction for every TB patient, can take up to 45 days or more, as many of us know about it. Now, if I break this process down, and I said that it is more affordable, if I break this process down, a CBNAT costs around 2,500 rupees, give or take. I might be slightly off, depending on how the patient is accessing the test, in which form. It can cost, I'm taking the lower limit of this, as around 2,500 rupees is the cost of CBNAT. Then there is the cost of LPA, which can be about 5,000 rupees. Then there is the cost of culture, which can be 12 to 13,000 rupees. At the end of it, we are able to get 13 antibiotic readout, which can take 15 to 45 days. The question that we keep asking ourselves is, what if we do CBNAT and get a genomic DST in a single test, which can not only give the information of those 13 antibiotics which we're talking about, but we can also get the information of another 5 or 6 antibiotics, almost 18 to 19 antibiotics that we can get the readout of. And that is a remarkable change in the way the world is looking at tuberculosis. This is as remarkable as the new antibiotics which have come into therapy. This is as remarkable as the short course therapy which has come out. This is as remarkable if there was a vaccine to be available for tuberculosis. The ability in science, the ability in diagnostics, the ability in medicine to be able to replace 5 to 6 different tests, to be able to reduce the cumulative cost of those tests, to be able to reduce the time taken of discovery for the right medicine by a cost much lesser than all of them put together, is a remarkable place where the scientific field has brought us to. This is what we are bringing to you as a product. Omega TB is Haystack Analytics' proprietary method for performing TB whole genome sequencing, and this is now available for everyone. We are able to partner with a large number of laboratories, and I will show you that how far we have been able to cover this country with this test. But what is worth appreciating in this report is this is probably the only report in which you can see 18 antibiotic names written. And that is the beauty of whole genome sequencing. We are able to target, to find out the drug resistance of 18 antibiotics. And the background of this report is even more remarkable, where none of this is based on a targeted mutation. This is based on sequencing, where every DNA is screened for 4 million data points. So if I were to have tuberculosis and I were to undergo TB whole genome sequencing, the bacteria from my TB would be sequenced. Around 4.4 million bases would be analyzed and will be given out on the resistance or sensitive for tuberculosis. One of the things about doing whole genome sequencing is that it can also detect non-tuberculosis mycobacteria, because if you can imagine, I am sequencing the whole genome. If there is a genome which is not mycobacterial in nature, which can actually be non-tuberculosis in nature, we will still be able to detect it and report it. And very interestingly, what we are also able to do is that we are able to detect the presence of co-infections or mixed infections. Now, these are the places where clinicians are looking to get early answers. In case of mixed or co-infections, where the rise and fall of disease is very imminent, where a patient might start responding to a disease, but to a therapy, but then not respond to it further because the resistant clones grow in the presence of those drugs. We can actually detect it on day one. We can actually be able to say that this patient has a mixed infection. This patient has non-tuberculosis mycobacteria and not tuberculosis. This patient has mycobacterium tuberculosis along with non-tuberculosis mycobacteria. All of this in one test. And that is the beauty of the science and research that the world has done. And I have been very, very fortunate to be part of this research for the past 15 years. And we are able to do that in a single diagnostic test using in molecular diagnostics. So some of the key questions that most clinicians would want to know, what does this test do? We are able to test for 18 antibiotics. The specificity is greater than 99%, almost reaching 100% there. The sensitivity across the drugs reaches from 85 to 98%. While not important for the clinician, we are able to reconstruct the phylogenetic analysis, which gives us the ability to discover new mutations and make it available for the next patient, and the ability to detect mixed infections and co-infections. And I think these are some of the these were few of the less appreciated factors of TB whole genome sequencing, at least on my table. I did not appreciate it hard enough because I didn't think that mixed infections was very important. I didn't think that detection of non-tuberculosis mycobacteria was very important. But once we've now done almost 15 to 20,000 genomes across India, we now know there is almost 3 to 5% non-tuberculosis mycobacteria in the TB patients that we are dealing with. And there's a significant amount of mixed infection that there is. We are hoping to publish much of this. I will be sharing some of the publications that we have already done. Now, one of the things what I was telling you telling about was our ability to not create a kit every time. So there is a rifampicin kit version 1, there is a 2 and 3, and then there are more drugs and there are more mutations. Because we are doing whole genome sequencing, if I have to add another gene, I just need to look for it using my software. I don't need to create a new kit. So the approvals that we get is for a single kit which does whole genome sequencing. This test is almost a perpetual test till such time that somebody in the world of science comes up with something better than whole genome sequencing, because we are literally using the method by which mutations are discovered to report for an individual patient, that too at a cost which is 50% lesser than what the patient would have to pay using current methods of microbiology, and at a turnaround time which is almost 80 to 90% faster than current microbiology. The list of genes that you see here are constantly increasing, and we have been able to not only make this technology available to a large number of diagnostic service NABL certified diagnostic labs in India, for them to offer to patients to almost 10,000 pin codes in India. As a result of which, I think we are very, very proud to be partnering with the National TB Elimination Program, DBT and BIRAC, to bring tremendous amount of knowledge to the world of tuberculosis. These are some of the papers that we have had, international and national. In fact, the validation of this test was done in this paper, what you're seeing in Journal of Global Antimicrobial Resistance, with the National Referral Lab at JJ Hospital. We have had more papers where we have we have disclosed new mutations that we have found in the population. As I was saying, if you look at the performance of the test, it has really exceeded some of the very old and well-established tests. I don't think there is any microbiology test which has a specificity and sensitivity consistently of 98, 99, 97% of the drugs that we are talking about. We have done large-scale comparisons of culture DST versus CBNAT, culture DST versus LP, and culture DST versus WGS. And what you see is that the WGS comparison with culture DST is at a 93, 94%, and this is increasing. And it is not surprising because that is what would happen if we sequence the whole genome. But that it is playing out in that way is really encouraging for us to use this technology more and more. So what is the process that we follow in the lab for the Omega TB? The sample is collected. We enrich the sample. Now, there are a lot of questions around this point, so I will take a minute to talk about it. So TB genome sequencing can be done direct from sample, but can also be done using an enrichment. And we have chosen this way because this is how world over people do it at Oxford and several different countries. And the reason they have chosen this way are two. One is that direct from sample molecular methods run a very high risk of finding nucleic acid which may not be from a live bacteria. And we know that this has a particularly high impact for tuberculosis, which is a very long drawn disease. And in this case, what happens is, given that we are looking at 3, 4, 5, 6, even 12 months of treatment, we need to be able to treat with antibiotics, which is the point of time problem for the patient. By doing a mild enrichment of the bacteria, of mycobacteria in culture, which takes about 5 to 7 days, we are 100% sure that we are reporting for live Mycobacterium tuberculosis in the sample from the patient. And second, this gives us the ability to have near reproducible results, whether it is zero microscopy on the slide, whether it is scanty, whether it is 1+, whether it is 2+, whether it is 3+, it doesn't matter to us, because we are able to get beyond that, beyond those limitations that we can only do genome sequencing for 3+, we can only do genome sequencing for 2+. How many patients that you come across who are likely to be drug resistant, who have 2+ and 3+ TB bacteria? They've already taken all kinds of antibiotics for which the sputum load is already low. And this is a very well-known aspect, at least in the Indian context and LMIC context. So this is the reason we have taken whole genome sequencing as a method. It might add 2 or 3 days more, but it is remarkably lesser than anything else out there. It is at least 20 to 30 days lesser than anything. After that, we actually process with the proprietary method of sequencing the whole genome using short read sequencing and using software, which is now published and validated. We are able to give an astoundingly high sensitivity, high specificity, high reproducibility report, along with very high depth of sequencing, almost near impossible to be able to miss out on any sensitivity that there is. And mind you, I spoke here about sensitivity and not resistance. While every molecular test is about our ability to predict drug resistance, when we have that amount of depth in sequencing, we actually have very high confidence when we call something sensitive. So compared to other methods of genome sequencing, such as pyrosequencing, targeted sequencing, and all of that, whole genome sequencing is actually right there in terms of turnaround time. It is cheaper than any of those methods. It is faster than any of those methods to get a much wider scale of drug resistance profile. And I can't think of a reason why we would compromise on sequencing when the gold standard is whole genome sequencing. Everything else is a lesser cousin. We are able to do whole genome sequencing cheaper than Pyro, cheaper than targeted, faster than most of the methods today, and be able to give way more comprehensive result than anyone can do. In fact, since 2021, when we were doing the validation, we have been able to bring the turnaround time in some cases to almost 7 days on whole genome sequencing. Currently, we provide a promised turnaround time of 2 weeks. But there are samples which we release reports much faster than that. And as you can see, by 2024, we are talking about our latest data. We are at almost nearing a turnaround time of molecular DX. How much time does it take to get GeneXpert plus LPA1 and LPA2? I'm sure few people will tell me it takes less than a day. But in most cases, we understand, and honestly to ourselves, it takes 3 to 4 days. I'm talking about 7 days for 18 antibiotics, along with all the mutations that there are in the world as of yesterday and not as of 3 years back. So we have done multiple, we continue to do this, compare with the current methods, because this is how we are able to see that we are not only, when we started, we were better than current standard of care. Today, we are going beyond. So the numbers that I showed you about 93%, they are sitting at 98%, 96%, 93% for against SLPA. And as you can see, the number of times a line probe assay is calling something sensitive when it is actually resistant is almost 17% in case of isoniazid, and is almost 11% for fluoroquinolone and 7% for SLI. And those are very risky propositions for a patient and a clinician to be able to invest time and money into a test which is 11% less sensitive than another test which is already available. And that is something we should try to move into, where we are using the best technology in the world, which the rest of the world is using. With that one additional step of 5 to 7 days of enrichment, we have now been able, and probably we are the only ones in the world who are consistently doing whole genome sequencing, not just for pulmonary tuberculosis, but also for extrapulmonary tuberculosis. We have samples in form of urine, we have samples in form of gastric, some samples, pleural fluid, pus. All of these samples we are now able to sequence and give drug resistance profiles of. And as I was saying, sorry for the mess on the slides. There were almost 100 cities on the slides here, which we are now covering. This is slightly old, about 6 months old now. We are doing a lot more than that, and all of this is because of the partnerships that we have that we have been able to create. I think I wouldn't be too wrong if I say that almost every national lab in the private sector and many of the public sector labs who are offering whole genome sequencing might as well be using Omega TB from Haystack. We have had the unique fortune of also having worked with some of the international labs in this. The results of our work now for the second year in a row has been selected in the Union Conference, which is the world's most premier conference for tuberculosis researchers, clinicians alike. What we are able to do is we are able to tackle pulmonary tuberculosis as well as extrapulmonary tuberculosis. We have 75% of the samples are beyond extrapulmonary tuberculosis, what we have here. There are several things that we are beginning to learn from the work that we have done till now. We now understand that there is a high amount of pre-XDR in the population that we're dealing with, and this is obvious because the healthcare community uses a new technology as the last resort technology. So most of the patients whom we are providing the report to are likely to be MDR anyway, and in that finding pre-XDR is not unexpected, and we are I think where we are able to make a difference is that we are able to find pre-XDR down to a level where for a pre-XDR patient, a doctor is able to give almost a personalized therapy, almost what is done in cancer today. It's a personalized therapy, it's a precision therapy for a TB patient at a cost which is lesser than any other test that we do. What has been very interesting over the years, as I was telling you, is we have found our fair share of non-tuberculosis mycobacteria and co-infections. I think there are some patients whom we have, whose life we have saved just by enabling the doctor to stop an anti-TB therapy. We all understand that anti-TB therapy is very difficult for a patient to continue with the amount of adverse reactions that there are. There are patients who actually did not have tuberculosis in them, and we were able to find other bacteria which very clearly could have presented with the same clinical prognosis as tuberculosis. The ability of the doctor to trust a report and change the therapy from a primary TB therapy to an antibacterial therapy has saved several lives, and probably has saved patients from reaching to tertiary care centers needing a lot more supportive and palliative care, which often comes with higher classes of drugs, anti-TB drugs, which are used. When we compare whole genome sequencing, when we compare Omega TB to the current other methods which are used and which I think I hope over the next few years are phased out in a logical way. When we compare it to culture DST, which can give a result of 13 to 15 antibiotics in 6 to 8 weeks, and Omega TB is able to give drug resistance prediction for 18 antibiotics in about 3 to 4 weeks. As I said, sometimes much earlier than that. This turnaround time itself is a matter of concern for all of us. But then, as I said, we would rather give a report of 100% sure alive bacteria that we are sequencing rather than anything else. A CBNAT, which would give you results for 1 to 2 antibiotics, and LPA, which would give you about 3 to 5 antibiotics. Omega TB, using whole genome sequencing, is able to provide drug resistance profiles of 18 antibiotics, and as I said, along with the ability to detect mixed infections. I think one of the most rewarding experience for me as a scientist to come back from Oxford and establish this in India has been this one slide, where I think prior to using whole genome sequencing, we always used to talk about drug susceptibility testing, DST, TB DST. We were always doing drug susceptibility testing, and we were always talking about drug resistance. We would do drug susceptibility testing and say there is so much of MDR, there is so much of pre-XDR, there is so much of XXDR. All those numbers would be given. The moment we started doing whole genome sequencing, which is a test for drug resistance, we actually are able to see some hope in terms of utility of other drugs, in terms of drug susceptibility. At a population level, as I said, we do have a biased population where most of the patients whom we sequence are likely to be retreatment cases, to be high-risk MDR cases, to be contacts of drug-resistant cases, are treatment failure cases. We see a high amount of drug resistance. That is not surprising. But what we see is even in them, there are almost 9 to 11 antibiotics which will work on them. There's almost no resistance for those antibiotics, at least in these patients. And that gives a tremendous opportunity of personalized therapy for a pulmonologist and a clinician. And this is something which is not new in India, where therapy is tailor-made to ensure the best result with the least amount of adverse reactions and the least cost. And the challenge is, the only reason we are not able to use many of these antibiotics is because we are not testing for it. If we test for it, then we know whether it is resistant or not. If we know it is resistant or not, then we can use it for therapy. If we use it for therapy, we are giving ourselves the best chance that of the 3, 4, 5 antibiotics, anti-TB drugs that has been given to a TB patient, none of them the patient is resistant to. That is the beauty of doing whole genome sequencing. It provides better outcome at lower patient cost. So I'm coming to the end of this talk, and I will come back to this TB algorithm. And the opportunity here for all of us is to decide for all of us, whether I am a patient, whether I am a clinician, whether I am a scientist taking a decision, how do we use whole genome sequencing? Should we take a high-risk drug-resistant patients through the journey of 7, 8, 9 tests and discovery of the right medicines, or should we ask for whole genome sequencing the day we ask for CBNAT? Can we ask for whole genome sequencing that same day along with the CBNAT? And I think that is where we think the world can move into. The world can move into a place where we use genome sequencing not just for difficult to treat diseases like cancer, but equally difficult to treat diseases like tuberculosis. And cost should not be the barrier for a disease which is as prevalent as tuberculosis. And I think at Haystack, we have been able to achieve that to a huge extent. I'm not saying we have achieved our final target. I'm sure we will get there as well. But as of today, TB whole genome sequencing using Omega TB with Haystack Analytics and our partners costs 50% less than culture-based detection of 13 antibiotics. And it certainly costs much lesser if we add up the cost of all the different tests. I don't think there is a better way in the world to detect heteroresistance. And I don't think there is a better way in the world where we can use a discovery platform for detection widespread in our movement to eliminate tuberculosis. I'm leaving this question out to all of you. Who is the test best for? My job and my passion is to bring the best test, the most sensitive test, the most specific test, the most reproducible test, the most dependable test at the least cost and the least turnaround time. I'm leaving it to our clinician friends to decide how they want to place it in their regular practice. From our end, we would like to support all of those initiatives. What we have seen, at least today, any suspected drug-resistant case, and I want to come back to that slide which I started with on the definitions of drug resistance. It doesn't matter if I'm suspecting MDR or pre-XDR or XDR or polyresistance or monoresistance. Even if there is one drug that the patient might be resistant to, we should be able to do the right diagnosis to give the right treatment. With that, I will open up the forum for question and answers if you have any.
Thank you, Anirvan. That was a wonderful presentation. And there are already 5 questions and a question in a chat box. I'll read that for you. And there are few in chat as well. So first question is from Pushpalatha Manjunath. So she's commenting.
I think this is very... Chaitali, even you are welcome to answer the question.
Yeah. So DNA extraction is highly challenging since MTB bacilli cell wall is lipid rich. So yes, there is a special, some tweaked protocol we are using, Pushpalatha. And yes, high GC content is the main reason why this DNA extraction needs to be tweaked to get a good quality and quantity which is required to pass the QC for whole genome sequencing. So it is a tweaked protocol, but yes, it's easy to perform as well. Another question is from Vaishali Kongare. She's asking about what expertise technician and what is STAT. So yes, it need expertise technician when it comes to culturing. So mostly we are sending, when we are culturing the sample, so it requires labs also to be furbished with the high-end equipments like MIGIT and the biosafety level safety, which can avoid occupational health hazard to the technician who are performing it. Turnaround time for this test is totally depending on when culture will flag. We'll get some amount of growth and culture when we can extract, but definitely we are lowered the turnaround time to 15 days. Then there is a comment which is from Pushpalata, and then Dr. Sarjan Shah is asking about culture is required for all the samples, and can you explain that in detail? So we are culturing, when Dr. Anirvan also briefly told about in his presentation, that we are enriching the sample. As in cultures, turnaround time starts when we are putting it into the midget. So if it is a sputum, definitely it will grow faster as compared to any extrapulmonary sample where the load of bacilli is really low. So yes, whatever samples we received in the lab, we do culture all those samples for whole genome sequencing. Nirmal Kumar Jain is asking about, can WGS/NGS can be used as a followers on test? So I'm expecting this must be in difference in between diagnostic and follow-up patients. So for follow-up, culture is in the algorithm currently, just to check whether still viable organisms are there or not. So any molecular diagnostic test is not in the algorithm to use as a follow-up test for the patients. Dr. Ashok Ratan has asked, and there are many thank you and presentation strikes for you, Anirvan. And Dr. Ashok Ratan's question was, how frequent has been histoplasma in TB negative sputum?
I think I have replied to him saying that I will get back to you on this.
Yeah.
It's just amazing. Dr. Ashok Ratan has been in every webinar that we have. Thank you, sir, for your guidance and your blessings.
So is Jaipur where we can send sputum sample for WGS? So yes, Dr. M.K. Jain from Jaipur are sending the samples, and we have completed around 95 to 100 samples from Jaipur so far in our database. So you can send the samples from Jaipur. This has been asked by Dr. Rakesh. And chat box is
question on, do you wait for culture to turn positive? So we run the enrichment of midget, and there is a definition of midget to turn positive. We actually don't wait for it to turn positive. We're looking for mild enrichment, and generally we look for some flaky growth in the midget, and that is enough for us to take it up. But many times this also results in turning positive. And as Dr. Chetali said, it is sample to sample, it varies. Most of you will understand the problems in tuberculosis with that. But technically, as far as the SOP is, we don't need the culture to turn positive. There's a visual inspection of the tube so that we can take the tube very early on for whole genome sequencing.
So next question is from Dr. I'm able to see her name. So if it's disease with mixed population of MTB, sensitive or resistant, how to interpret? So it's about mixed infection or mixed population.
So yeah, I have not read the question, but I heard what you said, Dr. Chetali. So the interpretation is that if it is a mixed infection, and so we are able to say mixed infection at a drug level, if those drugs are showing mixed calls, we consider the patient to be resistant for that drug. And that is the advantage of whole genome sequencing. We actually are able to know if these drugs cannot and should not be used. It is almost in line with the guidelines of TB diagnostics, where any test provides a resistance to an antibiotic, that is not going to be used. In whole genome sequencing, we just follow the same method.
Dr. Rakesh is asking about, it is necessary that samples should be positive before sending for whole genome sequencing, or like CBNAT, even negative sample can be sent for whole genome sequencing. So definitely, Dr. Rakesh, whatever where you feel the patient is clinically suspicious about the TB, all samples can be sent for whole genome sequencing, because any which ways we are culturing the samples first to get the good growth of the organism to send it for whole genome sequencing. If we haven't received the growth for in the particular time for that particular patient or the sample, then we declare it as a negative and send the report you across. So Pushpa Lata is asking about approximate cost, including transportation method of sputum sample. About cost, Anirvan.
I think we can actually discuss the cost offline. We hope that the logistics cost is the same as any other logistic cost for our partners. The cost will depend on with whom you are accessing the test from. As I said, we offer the test through many of our partners, and the range is between 6,500 rupees to 8,500 rupees in general, to the best of our understanding. But that is the cost to the patient of the TB test.
Our next question is from Vaishali. She is asking about how many samples can be processed at a time.
Yeah, I think I've given that. So because we're doing whole genome sequencing, we can process almost 1,000 plus samples. In genome sequencing, the challenge is not how many samples can we do. In genome sequencing, the challenge is always how few samples we can do. And that is where we have been able to manage this by partnering with a lot of diagnostic service providers. But genome sequencing is a highly scaled technology. I'm sorry for that answer of 1,000 plus. I know it is slightly sideways answer, but yeah, we can do a lot, actually more than 1,000 samples. There's one question on whole genome sequencing is TB. The TB elimination program does not recommend any genome sequencing method. However, the Niksha portal does take genome sequencing results as a diagnostic method. We hope that the National TB Elimination Program will pick up genome sequencing as well. And we have very good reason to believe they will bring it in the guidelines as soon as possible. Last 2 years, they've already mentioned it in their advisory that they have released. So,
also
yeah.
in India, TB report it is there from Central TB.
In the Central TB Division report, it is already there.
Next question is sending the DNA extraction from TrueNAT machine acceptable? So I don't think so. It will be acceptable. We are having some different protocol for that. Next is from Dr. Jyoti. Do you wait for culture, I guess, to be turned positive? This has been already applied. Yeah. Which NGS platform you're using for the test?
Short read sequencing. That is what we use for. So not only in TB, at any point of time, when we are looking for large number of mutations, whether it is TB or any other kind of genome sequencing, short read sequencing is the platform which is used globally and which is reproducible in nature. Long read sequencing for detection of large number of mutations currently with the error rates is challenging.
Next question is from Jyoti. She's asking about available in Tamil Nadu and what is the cost. So cost we had already discussed, Jyoti, and yes, it is available in Tamil Nadu. Doctor, we have the capacity to process 1,000 plus sample in one batch. Again, there is a cost, sensitivity in case of CSF. So
we don't know this answer yet, particularly in CFS. It's a CSF. It's a very good question, Pushpalata. Let me see if I can find that out and reach out to you.
Next question is from Jyoti again. In which container should we collect the sample and how to transport? Same way as we are transporting the sample. It should be triple layer packing and tamper-proof container and in a cold chain. So that is for Jyoti. Again, for the sequencing platform that we have already applied. Yeah.
I'll just take this question. This is a very interesting question from Ankur Gupta, Dr. Ankur Gupta. In what clinical situations WGS is not useful? What are the limitations of WGS? It's a very good question. I do not have a ready-made answer to this, so I'm just thinking aloud here. One situation where I know that WGS is not going to be useful if the disease progression is at a place where more than the pathogen itself, there's a very high immune response that the person is undergoing right now. At which point the patient might be under severe stress and is likely to be hospitalized even in critical care. But there's actually no bacteria to treat there, so whole genome sequencing will not detect anything, and it is not going to be useful. Other situations where I think whole genome sequencing is not useful would be that if I'm just thinking, if it is sensitive on even if it is sensitive on CVNAT, you would still want to do whole genome sequencing. This is what I can think of. Thanks for that question. I will think of a more detailed answer and reach out to you, Dr. Ankur Gupta.
What are the limitations of WGS in TB testing from Dr. Vivek?
Where is this question, Dr. Chatterjee?
It's in chat box.
So I'm just saying thanks to Dr. Pushpendra Singh. By the way, for people who don't know, Dr. Pushpendra Singh has done his TB research from Professor Stuart Cole's lab, who was the first one to sequence the TB genome. Really fortunate to have him in the middle today. Thanks, Dr. Pushpendra Singh, for your kind words and also joining in. WHO, what was the question? Okay, what are the limitations of WGS in TB testing? In TB testing per se, I don't think there are any limitations for whole genome sequencing, except if the quality of DNA is not good, if the reconstitution of, if the DNA is fragmented, the sequencing will not work very well. These are not problems that we face because we take the enriched culture. But if genome sequencing were done in any other way, those would be the problems.
So,
is decontamination necessary before enrichment? Yes, it's a normal process. Are you reporting heteroresistance? Yes, we are. But apologies for isn't culture the longest step in TB diagnosis? Wouldn't culture this? Absolutely, yes. I think Dr. Chatterjee has already answered that. So in our experience, and we have published this, in our experience, we have reported WGS of scanty samples, scanty microscopy samples. We have reported of 3+ samples. Our mean turnaround time is around 10 to 14 days. 14 days is the sweet time that we have. There are cases in which the culture doesn't grow. We rarely find a sample where it takes a long time to grow and we give a whole genome sequencing. So we do find it, but it is a challenging part. We haven't been able to solve it in a way which we can offer reproducibly. We are working on it. If there is something, we'll let everybody know about it.
Again, the question on same line from Pushpendra Singh. So decontamination. So decontamination is the process of a midget culture. So we are following the same. What is the cutoff for heteroresistant? Also, have you done WGS on 1+ samples?
Yeah, we have done WGS for scanty 0, 1+, 2+, 3+, EPTB, all kinds of samples we have done and reported fantastic results. Cutoff for heteroresistance globally is 20%. 20% allele cutoff is the cutoff for heteroresistance.
So that was the chat and question and answers we have finished with. Yes.
Fantastic. Dr. Chatterjee, I'll leave it to you to close the session.
Thank you very much, Dr. Anirvan, as well as all the attendees and the remaining panelists from Haystack Analytics. Request you to show the same interest for our upcoming webinars in the series. Thank you very much. I'm closing this session.